Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to tackle a practice problem set that relates to our lecture on enzymes. Let's go ahead and get started with problem number 1. Problem number 1 reads, consider a biochemical reaction A to B, which is catalyzed by AB dehydrogenase. Which of the following statements is true? Let's work through each answer choice. A says the reaction will proceed until the enzyme concentration decreases. This is not quite right. Enzymes are catalysts, meaning they speed up the rate at which equilibrium is reached, but they are not consumed in the reaction. Their concentration doesn't determine whether or not the reaction proceeds indefinitely. Reactions proceed until equilibrium is reached and not until the enzyme runs out. So this choice is incorrect. B says the reaction will be most favorable at 0 degrees Celsius. This is also incorrect. Enzyme catalyzed reactions typically increase in rate with increasing temperature, of course, up to a point. Most enzymes have an optimal temperature that's closer to physiological conditions, which is around 37 degrees Celsius in the human body. At 0 degrees, the reaction would be slower, not more favorable. So this answer is just not supported by enzyme behavior. C says a component of the enzyme is transferred from A to B. That would imply that part of the enzyme becomes part of the product, but that's not what enzymes do. Enzymes participate in the reaction, but they're not consumed and they do not become part of the product. They may temporarily bind the substrate or form an intermediate, but at the end of the reaction, the enzyme is released unchanged. So C is incorrect. D says the free energy change of the catalyzed reaction is the same as the uncatalyzed reaction. This is correct. Enzymes do not affect the thermodynamics of a reaction. They lower the activation energy, which changes the rate at which the reaction happens, but they do not change the overall free energy. The delta G remains the same whether or not an enzyme is present. All the enzyme does is help the reaction get to the product state faster. So for problem number 1, the correct answer is D. Problem 2 says, which of the following statements about enzyme kinetics is false? Once again, let's read through each answer choice. A says an increase in the substrate concentration at constant enzyme concentration leads to proportional increases in the rate of the reaction. Now, this statement might sound reasonable at first, but it's actually false, and here's why. In enzyme kinetics, we know that increasing substrate concentration does increase the reaction rate, but only up to a point. When substrate is low, adding more does speed things up because more enzyme active sites are being occupied. However, once the enzyme becomes saturated, meaning every active site is already occupied, adding more substrate doesn't increase the rate any further. The reaction rate levels off and it approaches a maximum known as Vmax. So the increase in rate is not proportional at high substrate concentrations. The relationship is hyperbolic, not linear. So this makes answer choice A the correct answer to this question because it is the one that is false. But we're still going to work through the other answer choices. B says, most enzymes operating in the human body work best at a temperature of 37 degrees Celsius. This is true. That's normal body temperature, and most enzymes found in humans are optimized for those conditions. C says, an enzyme substrate complex can either form a product or dissociate back into the enzyme and substrate. This is also true. Once a substrate binds to an enzyme, the enzyme substrate complex is formed, but it's not guaranteed that a product will be made. The substrate can still unbind and revert back to its original form, so this description is accurate. D says, maximal activity of many human enzymes occurs around a pH of 7.4. This is also true. pH 7.4 is roughly the pH of blood and many tissues, and most human enzymes are optimized for this neutral environment. There are exceptions, of course, like pepsin in the stomach, but the general rule holds. So to wrap up, the false statement is answer choice A, because enzyme kinetics does not show a proportional relationship between substrate concentration and reaction rate across all concentrations. The curve levels off as the enzyme becomes saturated. Let's go ahead and move on to problem 3. Problem 3 says, some enzymes require the presence of a non-protein molecule to behave catalytically. An enzyme devoid of this molecule is called a blink. This is a simple definition problem. An apoenzyme refers to the inactive form of an enzyme that is missing its required cofactor or coenzyme. Without that non-protein helper molecule, the enzyme can't catalyze its reaction. So the correct answer here is B. Problem 4 says, which of the following factors determine an enzyme specificity? Let's work through the answer choices. A says the 3D shape of the active site. And this is absolutely correct. Enzymes are highly specific, and that specificity is dictated by the unique three-dimensional structure of their active sites. The active site has a very particular shape and chemical environment that matches only specific substrates, and this is sometimes described as a lock and key or induced fit model. So this feature is what truly determines which substrate an enzyme can recognize and act on. So 4 is A. We're still going to work through B, C, and D. B says the Michaelis constant, or Km. While Km does tell us about an enzyme's affinity for its substrate, it's just a measure of activity, not something that determines specificity. It describes how tightly the enzyme binds, not which substrate it prefers. So this is not the right answer. C says the type of cofactor required for the enzyme to be active. Now, while cofactors are essential for the function of many enzymes, they don't determine specificity for substrates. The cofactor might help with catalysis, but it doesn't dictate what substrate binds in the first place. So this isn't the correct answer either. D says the prosthetic group on the enzyme. Again, prosthetic groups are non-protein components that assist in enzyme activity, but they don't determine specificity either. They're usually involved in the reaction mechanism and not the substrate selection. So this is incorrect as well. So again, the correct answer here is A. The 3D shape of the active site determines an enzyme's specificity. Let's move on to problem 5. Problem 5 says, human DNA polymerase is removed from the freezer and placed in a 60 degrees Celsius water bath. Which of the following best describes the change in enzyme activity as the polymerase sample comes to thermal equilibrium with the water bath? A says increases then decreases. B says decreases then plateaus. C says increases then plateaus. And D says decreases then increases. At first, the enzyme is too cold to function efficiently. As it warms up, the molecular motions increase, and so does the rate of reaction. That's why the activity increases initially. But there's a limit. Enzymes are proteins, and if the temperature goes too high, the enzyme begins to denature. For human enzymes like DNA polymerase, that usually starts to happen significantly above 40 to 45 degrees Celsius. Since the water bath is at 60 degrees Celsius, that is well above the enzyme's optimal temperature. So, as the polymerase reaches that high temperature, it starts to lose its structure and function, and its activity decreases sharply. So the correct answer here is choice A. The activity increases at first, then it decreases as the enzyme denatures at the elevated temperature. Problem 6 says, in the equation below, substrate C is an allosteric inhibitor to enzyme 1. Which of the following is another mechanism necessarily caused by substrate C? So this question asks about a pathway where substrate C acts as an allosteric inhibitor to enzyme 1, which catalyzes the first step in the sequence. That means as more C is produced, it goes back and it inhibits its own production by binding to enzyme 1 at a site other than the active site. So this is a textbook example of negative feedback, also known as feedback inhibition. So the correct answer here for 6 is D. Negative feedback, as a friendly reminder, is a regulatory mechanism where the end product of a pathway inhibits an earlier step to maintain balance and prevent overproduction. In this case, substrate C shuts down the activity of enzyme 1 to limit its own synthesis. That's what makes choice D the correct answer. The other choices don't apply. Competitive inhibition would involve binding at the active site, not allosterically. Irreversible inhibition would permanently disable the enzyme. And feedback enhancement would mean C increases activity rather than suppressing it. So only choice D matches the described scenario. Let's move on to problem 7. 7 says the activity of an enzyme is measured at several different substrate concentrations, and the data are shown in the table below. The Km for this enzyme is approximately blank. To determine the Km of an enzyme from this table, we look for the substrate concentration that gives a velocity equal to half of Vmax. The Vmax here appears to be around 100 millimole per second, since that's the plateau the velocity reaches at high substrate concentrations. Half of Vmax would be 50 millimolar per second. From the table, we see that the reaction velocity is exactly 50 millimolar per second when the substrate concentration is 0.5 millimolar. So that tells us that the Km is approximately 0.5 millimolar, and the correct answer is A. With that, let's move on to problem 8 and 9. Consider a reaction catalyzed by enzyme A with Km value of 5 times 10 to the negative 6 molar and a Vmax of 20 millimolar per minute. Problem 8 says at a concentration of 5 times 10 to the negative 6 molar substrate, the rate of the reaction will be blank. If the substrate concentration is exactly equal to Km, then by definition, the reaction rate is half of Vmax. We're given that Vmax is 20 millimolars per minute. Half of that would be 10 millimolar per minute. So the correct answer for 8 is A. 9 says at a concentration of 5 times 10 to the negative 4 molar substrate, the rate of the reaction will be blank. Now, the substrate concentration is 5 times 10 to the negative 4 molar. This is 100 times greater than Km. When substrate concentration is much greater than Km, then the enzyme is saturated and the rate approaches Vmax. That means the rate is approximately equal to 20 millimolar per minute, and the correct answer for 9 is C. Let's move on to problem 10. Problem 10 says the graph below shows kinetic data obtained for flu virus enzyme activity as a function of substrate concentration in the presence and absence of 2 antiviral drugs. Based on the graph, which of the following statements is correct? A says both drugs are non-competitive inhibitors of the viral enzyme. B says Tamiflu increases the Km value for the substrate compared to Relenza. C says Relenza increases the Vmax value for the substrate compared to Tamiflu. And D says both drugs are competitive inhibitors of the viral enzyme. Now, looking at the graph, we notice that this is a Line-Weaver-Burke plot. So what we're actually looking at is 1 over velocity versus 1 over substrate concentration. That means that the x-intercept tells us about Km, and the y-intercept tells us about Vmax. Now, if the x-intercept moves closer to 0, that means that Km is increasing. And then if our y-axis moves up, that tells us that Vmax is decreasing. So keeping that in mind, let's look at Tamiflu. Notice that the line intersects the y-axis at the same point as the uninhibited enzyme. That tells us that Vmax is unchanged. But its x-intercept is different. The x-intercept is shifted to the right, closer to 0. So Km has increased. This pattern is characteristic of competitive inhibition. Now let's take a look at Relenza next. Relenza, its line has a higher y-intercept than the uninhibited enzyme, meaning Vmax has decreased. But its x-intercept hasn't changed, so Km is the same. That's what we would expect for non-competitive inhibition. But here's the thing, the question isn't asking us to categorize the drugs. It's asking which statement is correct based on the graph. And out of the 4 options, only B is true. Tamiflu increases the Km value for the substrate compared to Relenza. So 10 is B. Problem 11 says, the conversion of ATP to cyclic AMP and inorganic phosphate is most likely catalyzed by which class of enzymes? Let's really think about this. We're converting ATP into cyclic AMP and inorganic phosphate. So we're not forming a bond between 2 new molecules, so that rules out ligase. We're also not using water to cleave a bond, so it's not hydrolase. And we're not transferring a functional group from one molecule to another, so we can also eliminate transferase. What's really happening here is that a single molecule ATP is being rearranged into a new form, cyclic AMP, with the loss of a phosphate. This is a classic job for lyase. Lyases catalyze reactions where a molecule is broken down or rearranged without water or redox chemistry involved. So the best answer for problem 11 is C. Problem 12 says, which of the following is not a method by which enzymes decrease the activation energy for biological reactions? Let's work through our options. A says modifying the local charge environment. That's true. Enzymes can stabilize charge transition states by altering the electrostatics around a reaction site. B says forming transient covalent bonds. That's also valid. Many enzymes temporarily bond to the substrate during the reaction to stabilize intermediates or assist in rearrangement. C says acting as electron donors or receptors. That's another common strategy. Enzymes often act as acids or bases, donating or accepting protons or electrons to move the reaction along. Choice D says breaking bonds in the enzyme irreversibly to provide energy. This is not correct. It suggests that enzymes break their own bonds irreversibly. That would destroy the enzyme itself. If you remember, enzymes are catalysts. They're supposed to be reusable, not sacrificed in the process. So D is not a valid mechanism, and it's going to be the correct answer here. 12 is D. Problem 13 says, a certain enzyme that displays positive cooperativity has 4 subunits, 2 of which are bound to substrate. Which of the following statements must be true? A says the affinity of the enzyme for the substrate has just increased. B says the affinity of the enzyme for the substrate has just decreased. C says the affinity of the enzyme for the substrate is half of what it would be if 4 sites had substrate bound. And D says the affinity of the enzyme for the substrate is greater than with 1 substrate bound. Since the problem tells us that the enzyme displays positive cooperativity, that means that when one substrate binds, it makes it easier for the next one to bind. Since the enzyme has 4 subunits and 2 are already occupied, we're somewhere in the middle of this cooperative process. That means that the enzyme's affinity for the next substrate is now higher than it was after the first substrate bound. So if we're looking at our options, we're not just thinking about whether affinity increased in general. We're specifically being asked about what must be true now that 2 subunits are bound. And the best answer here is going to be D. It says that the affinity is greater than when 1 substrate is bound. And that's a perfect fit for the definition of positive cooperativity. As more subunits bind substrate, the remaining ones bind more easily. So 13 is D. Problem 14 says, which of the following is least likely to be required for a series of metabolic reactions? A says triacylglycerol acting as a coenzyme. That's the one that should catch our attention right away. Triacylglycerols are fats that are used for long-term energy storage, not for directly participating in metabolic reactions as coenzymes. Coenzymes are usually small organic molecules like NAD+ or FAD that assist enzymes. But triacylglycerols are way too big and not functionally suited for that role. So this is the answer choice that's correct for problem 14, but we're still going to work through B, C, and D. B says oxidoreductase enzymes. These are commonly used in metabolic pathways for oxidation reduction reactions. C says magnesium acting as a cofactor. Magnesium is a well-known cofactor for enzymes, especially those involving ATP. And D says transferase enzymes. These are enzymes that transfer functional groups from one molecule to another, and they're a very common step in metabolic sequences. So putting it all together, the correct answer here is A. Triacylglycerol is least likely to act as a coenzyme in metabolism. With that, we can move into our last and final problem, which says, how does the ideal temperature for a reaction change with and without an enzyme catalyst? A says the ideal temperature is generally higher with a catalyst than without. B says the ideal temperature is generally lower with a catalyst than without. C says the ideal temperature is characteristic of the reaction, not the enzyme. And D says no conclusions can be made without knowing the enzyme type. Now, enzymes are proteins, and like most proteins, they have an optimal temperature range where they function best. If you go too high, the enzyme denatures and it loses its shape and therefore its function. Now, in contrast, an uncatalyzed reaction doesn't have to worry about enzyme structure. Higher temperatures generally increase the rate of those kinds of reactions because more molecules have enough energy to overcome the activation barrier. So if we compare the two, the ideal temperature for an enzyme catalyzed reaction is generally lower than that of the same reaction without an enzyme. And that's because the enzyme lets the reaction proceed quickly at a more moderate temperature, whereas the uncatalyzed version would need more thermal energy to move forward at the same rate. So the correct answer for problem 15 is B. And with that, we've completed this problem set. I hope it has been helpful. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.